Apple Podcast title, The Deep Dive, Nature's Cheat Code, How Life Hacks the Deep Freeze. Apple Podcast description. Have you ever wondered how fish survive in freezing Arctic oceans, or how an Alaskan beetle withstands negative 30 degrees? In this episode of The Deep Dive, we unpack the miraculous world of antifreeze proteins, AFPs, and ice-structuring proteins, ISPs. We explore the evolutionary heists, the quantum-level physics of thermal hysteresis, and the mind-bending biology that prevents living cells from turning into solid ice. Plus, discover how these same biological cheat codes are sitting in your kitchen freezer right now, and how they might just be the key to human cryopreservation and space exploration. Tune in for a fascinating journey through extreme survival, convergent evolution, and the future of cryobiology.
Have you ever left a glass bottle of water in the freezer and just completely forgotten about it?
Oh, definitely.
Right, and you come back a few hours later, maybe the next morning, and you open the freezer door to an absolute disaster.
It's just shattered glass everywhere.
Everywhere. The water inside froze, it expanded, and it just violently destroyed its container.
Because water expands when it turns to ice. It's just a fundamental rule of nature.
Exactly. Now, I want you to take that exact same scenario, but I want you to shrink it down to the microscopic level.
Okay.
Imagine those sharp, jagged, expanding ice crystals forming not inside a rigid glass bottle, but inside the incredibly delicate, microscopic cells of a living organism.
That sounds like a nightmare.
It really is.
Yeah.
By all the laws of biology, physics, and chemistry, it should be an absolute death sentence.
The physical expansion alone would just rupture the cell membranes,
Shred the internal organelle.
systematically destroy the tissue from the inside out.
Right. It should be game over.
Yeah.
And yet, if you look at our planet, life is incredibly, almost absurdly stubborn.
It really is.
I mean, life absolutely thrives in the freezing, lightless oceans of Antarctica. It thrives beneath miles of pack ice in the Arctic, and it survives the bitter subzero winters of the Northern Hemisphere.
It's a fundamental biological paradox. It's puzzled scientists for generations, honestly.
Because they should just freeze.
Exactly. We are talking about environments where the ambient temperature drops well below the freezing point of water. And since living things, from a microscopic algae to a massive fish, are largely made of water.
They're essentially just big bags of water.
Right, so they should theoretically freeze solid the moment the temperature dips. Their blood should turn to slush. Their cells should crystallize, but they don't. And the way they avoid this seemingly inevitable biological fate is nothing short of an evolutionary miracle.
And that brings us to the mission for today's deep dive. We are going to discover the biological cheat codes that allow life to survive the deep freeze.
I like that. Cheat codes.
It really feels like they are hacking the system. Our source material for this exploration is a really comprehensive look into a miraculous class of molecules known as antifreeze proteins.
Or AFPs for short.
Yes, AFPs. Though, as you'll find out a bit later today, if you look at a food label, they are sometimes more commercially known as ice-structuring proteins.
Which is a brilliant rebrand.
It is. Okay, let's unpack this. Because when I first started reading through these sources, I thought this was just about a few weird fish. But how big is the scope of this phenomenon, really?
The scope is massive. And frankly, it spans the entire tree of life.
Wow.
In this deep dive, we are going to explore the literal physics of freezing and how these incredibly specialized proteins hack those physics.
We'll look at the wild evolutionary leaps that created them, too.
Exactly.
Sure.
Sometimes seemingly out of nowhere or through methods that defy our traditional understanding of genetics. We will examine the surprising and, frankly, beautiful diversity of these molecules across fish, insects, plants, fungi, and microbes.
It's everywhere.
And finally, we will look at how these very same proteins have made the leap from the freezing Arctic oceans into commercial, everyday applications.
Which is my favorite part.
There is a very good chance that some of the mechanisms we discuss today are sitting right inside your kitchen freezer right now.
I absolutely love that. It takes something so alien and just brings it right into your home. So I want to start by redefining what we mean by the word antifreeze.
That's a good place to start.
Because I think for you, me, and anyone listening, when you hear that word, your mind immediately goes to the garage.
Right, car maintenance.
You think of that brightly colored neon green or pink toxic liquid you pour into your car's radiator before the winter hits.
Which is usually a chemical called ethylene glycol.
And it works by what chemists call colligative properties.
Which is a term that sounds a bit intimidating.
It does.
But it essentially means it works by sheer brute force volume.
Right. I was reading that with car antifreeze, You literally have to pump a ton of it into the water to get the freezing point to drop.
You're fundamentally changing the ratio of the liquid.
It's like trying to stop a crowd from dancing by just shoving an equal number of people who refuse to dance into the room.
That is a brilliant way to put it. It's a volume game.
Just crowding the floor.
And if we connect this to the bigger picture of a living, breathing organism, a brute force approach just isn't biologically viable.
Because you can't just fill a fish with neon green chemicals.
Exactly. You cannot simply pump a living fish full of a traditional solute like ethylene glycol. And even if it wasn't toxic, you couldn't pump them full of massive amounts of salt or sugar to lower their freezing point either.
Because it would completely mess up their internal balance, right?
Yeah.
I mean, fish already have to regulate their salt levels so carefully.
Exactly. It would cause a massive fatal shift in osmotic pressure.
Osmosis being the water balancing act.
Right. Osmosis is the process that dictates that water always wants to balance out solute concentrations across a cell membrane.
Okay.
If a cell suddenly floods its interior with massive amounts of a solute, like sugar or salt, just to prevent freezing, the water outside the cell would rush in to dilute it.
And the cell would swell up.
And literally burst. Or, conversely, if the solute was heavily concentrated in the blood, it would draw all the water out of the surrounding tissues, completely dehydrating the animal from the inside out.
So the animal would basically turn into a piece of biological beef jerky.
Precisely. They would die of dehydration while surrounded by water.
That is a terrible irony.
So the absolute beauty of antifreeze proteins is that they work in a non-colligative manner.
Meaning they don't rely on sheer volume at all.
Not at all.
I was floored by the numbers in our sources. They note that AFPs are effective at concentrations 1/300th to 1/500th of other dissolved solutes.
It's a fraction of a fraction.
If we go back to our room full of dancers, instead of flooding the room with non-dancers, the organism just sends in 2 or 3 highly trained bouncers to stand exactly where the dancing starts and shut it down.
I like the bouncer analogy.
It is a hyper-precise biochemical surgical strike.
They don't flood the system. They target the problem at incredibly low concentrations, which completely minimizes any dangerous effect on the organism's delicate osmotic pressure.
And the way those microscopic bouncers execute that targeted strike is through a fascinating physical mechanism.
They don't just float around randomly. They selectively seek out and bind to specific crystalline forms of ice the microsecond they begin to form,
Just grabbing them.
essentially creating a physical blockade that stops the ice nucleation process dead in its tracks.
And this leads us to a term in the source material that I absolutely have to geek out over.
Oh, I know which one you mean.
The term is thermal hysteresis. I'm not going to lie, it sounds like an incredible sci-fi band name.
It really does.
But it is actually the core fundamental mechanism of how AFPs keep these creatures alive.
It is a great term, and it is the key to everything we are talking about today. Hysteresis generally refers to a phenomenon where the state of a system depends on its history, or where there's a lag in a reaction.
Okay.
To break thermal hysteresis down for you in this context, it is effectively the artificial gap created between the melting point of water and the freezing point of water.
Right, because normally those two things are the exact same temperature. Water freezes at 0 degrees Celsius and ice melts at 0 degrees Celsius. It's a two-way street at the exact same intersection.
Exactly. Under normal thermodynamic laws, it's a perfectly balanced equilibrium.
But when an organism possesses antifreeze proteins, it breaks that equilibrium.
Those proteins physically wedge themselves at the microscopic interface between the solid ice crystal and the liquid water surrounding it.
And by doing this, they kinetically inhibit the growth of the ice crystal.
They push the freezing point down while the melting point remains exactly the same.
I love the phrasing in the source material. It stops the thermodynamically favored growth.
Yes.
Physics says, I'm at 0 degrees, I want to freeze, I need to freeze.
And the protein steps right in the middle of that chemical reaction and says, access denied.
That is a perfect way to visualize it. The proteins literally cover the water-accessible surfaces of the microscopic ice crystals. They hold open this gap, this hysteresis.
They create a state of tension where the water should be ice, but it is forced to remain liquid.
And looking at the data from the sources, the maximum level of thermal hysteresis shown by fish AFPs is approximately negative 3.5 degrees Celsius.
Which is about 29.3 degrees Fahrenheit.
Now, when I first read that, I thought, wait, negative 3.5 degrees? That doesn't sound like a massive drop.
It doesn't sound like much.
I was expecting them to push it down to negative 50 or
Are
something.
they sure?
But then the material clarified why that is exactly perfectly what they need.
Right, because aquatic organisms are typically only exposed to water temperatures that drop to about -1 or -2 degrees Celsius below freezing.
Because ocean water itself generally freezes around -1.9 degrees Celsius.
Mm-hmm.
The salt content lowers the freezing point of the ocean itself.
Exactly. So if the fish's blood is protected down to -3.5, that gives them an absolutely perfect, comfortable safety buffer.
But there is a catch here, isn't there?
Yeah,
It's not a flawless magic shield.
This raises an important question about the limits of biology, and you are right to point it out. Yeah.
The catch is that the rate of cooling absolutely matters. These proteins are incredible marvels of engineering, but they are still physical molecules floating in a fluid.
They need time to work.
They require a little bit of time to physically find the nascent ice crystals, orient themselves, and bind to them.
So if there is a sudden rapid plunge in temperature.
It can actually substantially decrease the thermal hysteresis value.
Oh, wow.
The freezing point drops so fast, and the ice grows with such violent speed that the proteins simply cannot keep up.
They are overwhelmed.
Consequently, these organisms can still easily freeze to death if the temperature drops too abruptly.
So they need the cold to arrive at a relatively natural, gradual pace.
Yes, so their internal defenses can mobilize and cap the ice crystals as they form.
So it is this dynamic, high-stakes battle between the speed of the temperature drop and the physical reaction time of the proteins.
It's a race.
That is incredibly dramatic when you think about it happening inside the blood vessels of a tiny fish. And it perfectly sets up something else I found fascinating in the sources. Which is?
Not every organism uses these proteins the exact same way. The material outlines two very distinct survival camps. I'll admit, one makes total sense to me, and the other sounds completely insane.
I think I know where you're going with this.
Can you clarify the difference between the freeze-avoidant species and the freeze-tolerant species?
Gladly. The first camp is the freeze-avoidant species, which is the one that probably makes the most intuitive sense.
Right.
These are the organisms, like the fish we just discussed, that use AFPs to prevent their body fluids from freezing entirely.
Their singular goal is zero internal ice.
They hold the line, they use the proteins to create that thermal hysteresis gap, and as long as they stay within that temperature buffer, they remain completely liquid.
But as we just discussed with the cooling rates, if the temperature drops too low or too fast, the AFP function is overwhelmed, ice grows rapidly throughout their body fluids, and they die.
Yes, for them, any significant internal freezing is fatal. It's an all-or-nothing defense mechanism.
The shield either holds or the organism perishes.
But the second camp, the freeze-tolerant species,
This is the one where my mind just gets blown.
it is truly remarkable.
Ugh.
Freeze-tolerant species actually survive the freezing of their internal body
They
fluids.
don't just tolerate the cold, they tolerate turning into ice.
For these organisms, the AFPs aren't trying to stop the freeze entirely. They aren't trying to maintain a purely liquid state.
So what are they doing?
Instead, they act as what we call cryoprotectants.
Meaning they manage the freezing process rather than preventing it?
Exactly.
But how does that not shred their cells like the glass bottle in the freezer?
Because they inhibit something called recrystallization.
Recrystallization.
When ice first forms, it usually starts as lots of very tiny microscopic crystals. On their own, these tiny crystals aren't immediately lethal,
Okay.
but over time, thermodynamics pushes those tiny crystals to fuse together into larger, much more stable, and much more destructive ice crystals.
It's the large crystals that act like microscopic knives, slicing through cell membranes and destroying tissue. Okay, so the AFPs in freeze-tolerant species, they bind to the tiny crystals and stop them from fusing together.
Yes, they keep the ice small and manageable. This stabilizes the cell membranes and prevents the massive tissue damage.
They basically turn the deadly knives into harmless sand.
That is exactly it. What's fascinating here is that to pull this off, these freeze-tolerant organisms often work in conjunction with another type of protein altogether.
Called ice-nucleating proteins, or INPs.
Yes.
Now wait, I had to read this twice in the notes. They actually have a protein specifically dedicated to starting the freezing process. Why would they want to do that?
It sounds counterintuitive, but it's a stroke of genius. The INPs intentionally trigger the freezing process at relatively high subzero temperatures.
Oh, because if they control when it freezes, they control how it freezes.
Exactly. They take control of the environment. By initiating the freeze early and predictably, it allows the organism to control the rate of ice propagation.
It's a slow, managed rollout of ice.
The INPs start the fire, so to speak, in a controlled manner, while the AFPs ensure the resulting ice crystals stay incredibly small and harmless.
It is a highly choreographed, biologically controlled freeze that allows the organism to survive, being effectively frozen solid for months at a time.
It's brilliant biological engineering. They aren't fighting nature.
They are conducting it. So now that we have the physics down, the hysteresis, the bouncers at the door, the managed freezing, let's dive into the animals themselves.
Let's do it.
Because the history of how we even figured this out is a great story. Let's set the scene. It is the 1950s. We have a Norwegian scientist named Skalander.
A pioneer in the field.
He is out on boats in the freezing, brutal conditions of the Arctic. He is pulling up these fish from the dark water, and he is just absolutely baffled.
Because they should be dead.
He is taking temperature readings, and the water is definitively colder than the known freezing point of the fish's blood. By all rights, the moment these fish are pulled out, or even while they are swimming, they should be solid blocks of ice floating in the sea. He suspects there must be some sort of antifreeze dissolved in their blood, but the analytical tools of the 1950s just aren't quite there to let him pin it down. He knows it's happening, but he can't prove how.
It was an incredible biological mystery.
It really was.
It wasn't until a couple of decades later, in the late 1960s, that an animal biologist named Arthur de Vries finally managed to isolate the culprit.
And he was working in the Antarctic, right?
He was. And through rigorous chemical isolation techniques, he discovered a unique class of molecules in their blood that he called antifreeze glycoproteins, or AFGPs.
Antifreeze glycoproteins. And this discovery completely changed how we viewed cold weather biology. But my favorite part of the source material is why these fish had them in the first place.
Yes, and if we connect this to the bigger picture, to understand why de Vries found these highly specialized proteins in Antarctic fish, we have to zoom way, way out.
Like geological timescales.
Exactly. We have to look back at the geological and climatic history of the entire planet. The sources detail some incredible data pulled from deep-sea ocean drilling cores.
Those are the literal cylinders of Earth pulled from the ocean floor.
Yes, that allows scientists to read the history of the planet's climate like the rings of a tree.
And those cores reveal an incredible dramatic story.
Over 30 million years ago, a massive, planet-altering geological shift occurred. The continents were shifting, and this led to the formation of the Antarctic Circumpolar Current.
This is that massive, incredibly powerful ocean current that constantly, relentlessly circles the entire continent of Antarctica, right?
It is, and when that current formed 30 million years ago, it effectively created a dynamic physical wall around Antarctica.
Wow.
It isolated the continent and its surrounding waters from the warmer, more temperate ocean currents of the rest of the globe.
It basically cut off the global central heating.
Exactly. This initiated a massive planetary cooling event in the southern hemisphere. The oceans around Antarctica plummeted in temperature, turning into the freezing, hostile environments we know today.
And what happened to the life there?
Well, as a result of this sudden, in geological terms, deep freeze, there was a mass extinction. The vast majority of teleost species,
Which are the ray-finned fishes.
yes, the ray-finned fishes that populated those waters, simply couldn't withstand the freezing temperatures. Their blood crystallized, their populations were decimated.
It was an icy apocalypse for the fish down there, but not all of them.
No. A specific, seemingly unremarkable suborder of fish called the notothenioids managed to survive.
And why did they survive?
Because by pure evolutionary chance, they had evolved this precise genetic adaptation, the antifreeze glycoprotein gene.
Just a lucky roll of the genetic dice.
While the rest of the fish died off in droves, the notothenioids didn't just scrape by, they thrived.
They diversified. They evolved into numerous different species and essentially took over all the newly emptied ecological niches left behind by the mass extinction.
It's the ultimate evolutionary success story.
They basically looked around and said, oh, the water is freezing and everyone else is dead? Great, more real estate for us.
They inherited the entire bottom of the world because of one genetic quirk.
But here's where the genetics get really, truly crazy. Our notes highlight a mind-bending fact about convergent evolution.
This is one of my favorite examples.
We just talked about the Antarctic notothenioids down at the South Pole 30 million years ago. Now, I want you to look all the way up at the North Pole at a fish called the Arctic northern cod.
Right.
These two groups of fish are separated by the entire planet. They live in completely different oceans. They diverged from each other evolutionarily somewhere between 7 to 15 million years ago.
They are not closely related at all.
And yet, scientists look at their blood and they both have nearly identical antifreeze glycoproteins.
This is a textbook prime example of convergent evolution.
Two completely unrelated species, separated by thousands of miles, facing the exact same brutal environmental pressure,
Freezing waters.
independently arrive at the exact same biochemical solution. It is nature finding the optimal answer to a problem twice.
But what is truly phenomenal, and what had geneticists reeling, is how they arrived at that solution. They built the exact same protein, but they used entirely different genetic blueprints and raw materials to do it.
All right, I was reading about where these genes actually came from. So the Antarctic notothenioids, they evolved their antifreeze gene from a digestive enzyme.
The material specifically calls it an ancestral pancreatic trypsinogen-like serine protease gene.
Which is a very long way of saying it was a gene responsible for making an enzyme that breaks down food in the fish's pancreas.
Exactly.
So somewhere along the line, over millions of years, a gene that was meant to help digest a meal accidentally duplicated, mutated, and somehow transformed into a gene that perfectly stops ice from forming in the blood.
That alone is wild.
But the northern cod in the Arctic, their evolutionary path was entirely different and much more recent. The sources say it happened only about 3.2 million years ago.
And their antifreeze gene didn't evolve from a pre-existing working gene like a digestive enzyme.
No. It emerged from non-coding DNA.
Yeah.
Non-coding DNA, which is what people sometimes colloquially call junk DNA, right? Just the biological filler material that doesn't seem to do anything.
Exactly. It was previously thought to be just evolutionary baggage.
But the cod's antifreeze protein emerged via tandem duplications of a very short sequence of this non-coding DNA.
Specifically, a tiny unit of amino acids. Threonine, alanine, alanine. It just duplicated over and over, right out of the dark, non-coding regions of the genome.
I saw a term in the material describing this, the proto-ORF model. And I will be honest, I need you to translate that into English for me because it sounds incredibly complex.
It is a cutting-edge concept in genetics. ORF stands for Open Reading Frame.
Okay.
It is effectively a section of DNA that has the potential to be read and translated into a protein. The proto-ORF model describes a very rare occurrence where a new gene essentially pre-exists as a fully formed, potentially useful sequence in the non-coding DNA.
It is just sitting there in the dark, silent.
Before the regulatory elements, the genetic on switches even exist to activate it.
So it's like a fully built, furnished house sitting in the middle of nowhere just waiting for someone to finally run a power line to it.
That is a brilliant analogy. The house is there, but the lights are off.
And then, boom, a massive environmental pressure like a rapidly cooling, freezing ocean comes along.
Through random mutation, a regulatory switch is flipped, the power line is connected, and this non-coding junk DNA suddenly illuminates and becomes a highly functional, life-saving protein.
So the cod literally hacked its own junk DNA to survive the freezing Arctic. Absolutely love that.
It's incredible.
But if you think that hacking your own DNA is wild, we have to talk about the smelt. Because we just discussed convergent evolution, where two animals invent the same thing independently. But there's another evolutionary curveball in the source material regarding a different type of antifreeze.
Type 2 AFPs.
These are found in 3 specific fish: the sea raven, the smelt, and the herring. Now, the notes point out a statistical impossibility. The smelt and the herring share up to 98% of the introns for this specific antifreeze gene.
Which, as you alluded to, is statistically impossible to achieve through independent convergent evolution.
We need to define introns quickly.
Introns are the non-coding sections of an RNA transcript. When a cell reads a gene to make a protein, it makes a rough draft of RNA, and then it literally splices out and throws away the introns before the final protein is made.
They're the cutting room floor scraps of genetics.
Precisely. Now, for two widely divergent lineages of fish, the smelt and the herring, to share 98% of these highly specific, seemingly useless cutting room floor sequences implies something much, much more direct than convergent evolution.
The smelt didn't independently evolve this gene over millions of years.
The scientific consensus, backed by undeniable genomic evidence, is that the smelt actually stole it from the herring.
I'm sorry, I need to pause here.
Yeah.
They stole a gene.
Yes.
Like a genetic heist.
Yeah.
How does a fish steal a gene from another fish?
It is a phenomenon known as horizontal or lateral gene transfer. We see it all the time in bacteria. It's how they share antibiotic resistance so quickly. But it is exceedingly rare in complex vertebrates like fish. The theory, which is heavily supported by the presence of transposable elements.
Basically jumping genes.
Exactly. Jumping genes in the smelt gene that are otherwise uniquely characteristic of the herring. The theory is that this transfer happened during mating.
Wait, herring and smelt don't mate with each other, do they?
No, but they do spawn in the same freezing, chaotic water columns. It is believed that during spawning, smelt sperm cells were somehow exposed to degraded or free-floating foreign herring DNA in the water.
Just floating around.
Through a microscopic stroke of unimaginable statistical luck, a smelt sperm cell absorbed that herring DNA, integrated the antifreeze gene into its own payload, and then successfully fertilized a smelt egg.
That is staggering. A microscopic sperm cell just bumps into some floating genetic debris from a completely different species, absorbs it, and suddenly the entire subsequent lineage of smelt has a survival superpower.
They literally acquired it horizontally across species lines.
It totally upends the idea of evolution being this slow, branching tree. It's more like a tangled web where sometimes you just grab a power-up from the guy swimming next to you.
It truly is a paradigm-shifting discovery in evolutionary biology. It shows how opportunistic life can be under extreme pressure.
Nature is absolutely wild. And just to show you, the listener, the sheer diversity of these proteins, the sources give us a roster of different fish AFPs.
We've talked deeply about the glycoproteins.
But there are 4 other completely distinct structural types of antifreeze proteins in fish. Let's run through them quickly, because I want to give a sense of how many ways nature solves this one problem.
Okay, first, there's type I.
This is found in the winter flounder and various sculpins. The material notes that this is the best documented one because it was the first to have its 3D structure fully mapped out by scientists. It looks like a single, long, amphipathic alpha helix.
Let's clarify that term, amphipathic, because it is crucial to how these proteins work. Amphipathic means the molecule has distinct faces with entirely different properties. Imagine a long cylinder. One side of the cylinder is highly hydrophobic, meaning it actively repels water. The opposite side is hydrophilic, meaning it attracts water.
Got it.
And nestled within this structure is a highly specific face made of threonine and asparagine amino acids. This exact architectural arrangement is what allows it to bind to ice, which we will detail much more closely when we look at the micromechanics.
Okay, so a two-phase cylinder. Then you have type I HIP, and HIP simply stands for hyperactive.
Found in right eye flounders.
And compared to the standard type I, it is a massive molecule. The notes say it's about 32 kilodaltons, making it a dimeric molecule.
Dimeric simply meaning it is composed of 2 identical halves, or monomers, bound together. It operates as a twin system.
Right. And because of this massive twin structure, it is incredibly effective at depressing freezing temperatures. It has multiple ice binding sites.
It's like having an antifreeze molecule with extra hands to grab onto the ice crystal from multiple angles.
Following that, we have the type 3 AFPs, which are found in the Antarctic eel pout.
These are fascinating because, similar to the digestive enzyme we discussed earlier, they evolved from a completely different functional gene.
In this case, a sialic acid synthase gene, or SAS.
Through a complex gene duplication event, this SAS gene actually lost its N-terminal part.
It basically lost a chunk of itself.
And the remaining fragment evolved into a highly compact, globular, highly effective antifreeze protein. It shows yet another completely unique evolutionary pathway.
And finally, type 4, found in longhorn sculpins. This one is an alpha helical bundle. It's incredibly rich in glutamate and glutamine amino acids. So, just stepping back for a second.
Yeah.
In fish alone, we have at least 5 completely distinct structural molecules. Glycoproteins, type I cylinders, type I hyperactive twins, type 3 globular fragments, and type 4 bundles.
They all look completely different.
They all evolve completely independently, and they're all designed to do the exact same thing: stop ice.
It's a testament to evolutionary pressure.
But as we pivot away from the ocean, we learn that the macrofauna, the big fish, are not the only ones playing this high-stakes game. We have to look at the microworld.
We have to look at the microbes, the fungi, and the fascinating paradox of plants.
Let's start with the sea ice ecosystem. When you look at a documentary about the polar ice caps, the ice just looks like dead, frozen, sterile water. But it's not.
It is absolutely teeming with life.
Diatoms, which are a type of microscopic algae, absolutely dominate the polar sea ice communities. Specifically, species like Fragilareopsis cylindrus.
Yes, and these diatoms possess AFPs that belong to a large, complex, multi-gene family. They need them because they actually live inside the tiny brine-filled channels within the sea ice itself.
But what is really intriguing about this specific family of ice-binding proteins is that it isn't just found in diatoms.
The exact same homologs are represented across wildly different, unrelated taxa in the sea ice ecosystem. You find variations of these ice-binding proteins in psychrophilic or cold-loving bacteria.
You find them in cold-tolerant fungi, like the snow mold fungi that survive under the winter-packed snow.
And wait, I have to bring this up because it shocked me. The notes say you even find them in edible mushrooms, like the enoki and shiitake mushrooms.
They absolutely do.
The enoki mushrooms I literally put in my ramen last week have antifreeze proteins.
They do. It's how they survive sudden frosts in their natural environments.
Wow.
And the fact that we find this same family of proteins in diatoms, bacteria, snow mold, and your dinner mushrooms points to one of two things.
What's that?
Either there is a deeply ancient shared ancestral origin for these proteins among all microorganisms dating back hundreds of millions of years, or
we are looking at an incredibly vast, complex network of horizontal gene transfer,
like the smelt in the herring,
occurring constantly in the microscopic sea ice community.
the idea of a global microscopic gene-sharing network under the ice is incredible. Okay, but my absolute favorite microbe from the source material has to be the ciliate consortium, Euplotes.
This little single-celled organism has an ice-binding protein called AFPs, and it is an absolute masterclass in freeze tolerance.
Is a perfect example of what we discussed earlier. Remember our distinction between freeze tolerance versus freeze avoidance.
The Euplotes protein doesn't focus entirely on creating a massive thermal hysteresis gap to prevent ice formation entirely.
Instead, it is exceptionally good at ice recrystallization inhibition.
It works efficiently all the way down to a staggering negative 7.4 degrees Celsius. It allows the ice to form around it, but perfectly manages the crystal size to prevent its single cell from being punctured.
And the science gets even cooler. Researchers actually took the specific gene for this Euplotes antifreeze protein, isolated it, and inserted it into the genome of standard E. coli bacteria in a lab.
And the result?
The newly modified E. coli survived significantly longer periods at 0 degrees Celsius. This little single-celled ciliate's genetic code is so robust, so perfectly engineered by nature, that you just plug it into a standard lab bacterium like a software update and instantly upgrade its cold resistance.
Which, as you can imagine, has massive implications for biotechnology and synthetic biology, which we will certainly get to.
But first, we need to address the plants. Because as the sources note, the classification of AFPs became much more complicated in the 1990s when antifreeze proteins were finally discovered in plants, specifically in winter rye leaves.
The plant exception.
So I'm picturing a field of winter rye covered in snow. What makes plant AFPs so fundamentally different from the fish and the microbes we've been talking about?
What's fascinating here is that plant AFPs, which are often classified by botanists as dehydrins, operate on a slightly different biological paradigm.
First off, if you measure them, they exhibit much weaker thermal hysteresis activity compared to the hyperactive fish or insect AFPs.
Their primary physiological function isn't really to prevent ice formation altogether. They know they're going to freeze.
They can't swim away to deeper, warmer water. They're literally rooted in the freezing ground.
Exactly. They are entirely immobile. So, like the ciliate we just discussed, they are heavily focused on managing the inevitable. They inhibit the recrystallization of ice.
They let the water in their cellular spaces freeze. But the dehydrins stop those ice crystals from growing large enough to shred the plant's delicate vascular system.
They are the elegant managers of the cold. They let the frost in, but they dictate the terms.
But the evolutionary origin of these plant proteins is what really caught my eye in the reading, because it's a completely different origin story than the digestive enzymes of the fish.
It is a brilliant, highly resourceful pivot by nature. Analysis shows that most plant AFPs actually evolved from pathogenesis-related proteins.
Pathogenesis.
Yeah.
Meaning proteins that originally evolved to fight off diseases.
Precisely. They originally evolved as defense mechanisms to fight off fungal infections and other pathogens.
And what is truly remarkable, what makes it a biological marvel, is that some of these plant antifreeze proteins still retain their original antifungal properties.
They are pulling double duty. They are protecting the plant from deadly, expanding ice crystals in the dead of winter, while simultaneously fighting off opportunistic fungal pathogens that might try to take advantage of the stressed freezing plant.
It's a 2-for-1 survival tool, a shield against both ice and disease. It is incredibly elegant. But if plants are the elegant managers of the cold, Insects are the absolute biological tanks.
Here's where it gets really interesting.
You think a fish surviving at -2 degrees Celsius is impressive. Our sources introduce us to the spruce budworm. During the extreme, brutal winter months in northern forests, this little moth larva resists freezing at temperatures approaching -30 degrees Celsius.
Just for context, that is -22 degrees Fahrenheit. It is unimaginably cold.
To survive temperatures that extreme, the proteins involved have to be incredibly robust.
Insect AFPs are generally referred to in the literature as hyperactive. They have a significantly greater thermal hysteresis value than most fish AFPs.
They have to, because the air temperatures they face are vastly colder than any liquid ocean.
Structurally, insect AFPs are highly repetitive. They form a shape that biochemists call a beta-solenoid, or a coil structure.
I was trying to visualize this. Imagine a tightly wound spring, like from a mattress, but instead of being perfectly round, one entire side of the spring is completely flattened out. That flat side is the ice-binding surface.
That is a highly accurate visualization, and nature has engineered several distinct variations of this flattened coil across different species of bugs. For instance, let's look at the tenebrio and dendroids beetles.
Their proteins are heavily reinforced.
They have internal disulfide bonds that act like microscopic structural braces, holding the coils rigidly together. And on that flat ice-binding side of the coil, they have a double row of an amino acid called threonine.
It's like armored plating.
Very much so. But then compare that to the radium beetles. Their proteins have even longer repeats. They don't rely on those internal disulfide bonds for bracing.
Instead, they form what is called a compressed beta sandwich structure.
And instead of a double row, they boast a formidable 4 rows of threonine residues on their ice-binding surface. They maximize the contact area with the ice.
And then you have the midges, which use a structurally similar solenoid coil, but their flat ice-binding surface consists of a single row of tyrosine residues instead of threonine.
It's exactly like looking at different models of armored vehicles. They all have the exact same goal, survive the battlefield of winter, but the armor plating, the internal bracing, the structural layout is arranged completely differently depending on the specific evolutionary path they took.
But then I asked about the snow fleas, because they are famous here in North America, especially in Canada.
You see them literally hopping around on the surface of the snow on relatively warm winter days.
They aren't actually true insects. They are springtails, which are a type of primitive six-legged arthropod. And their AFPs, despite living in the snow, are totally different from the beetles, right?
Completely different. The similarity entirely ends at the fact that they have a flat ice-binding surface. The snow flea AFP is roughly 50% composed of the amino acid glycine.
It doesn't form a beta-solenoid coil.
Instead, it folds into a dense bundle of what are called polyproline type 2 helices. And instead of using threonine or tyrosine to bind to the ice like the beetles and midges, its ice-binding face is dominated by small hydrophobic residues like alanine.
Again, convergent evolution. A totally different genetic path, a totally different molecular shape, but they still arise at a flat surface that perfectly sticks to ice.
But I want to drop a bombshell from the sources right now.
Please do.
Because we have spent this entire deep dive talking about antifreeze proteins, the P in AFP, amino acids folding into complex shapes. But the Alaskan beetle, scientifically known as ceramboids, has an antifreeze that isn't even a protein at all.
This was a landmark discovery published in the Proceedings of the National Academy of Sciences in 2009.
It challenged everything we thought we knew about cryobiology. The Alaskan beetle produces a hyperactive antifreeze molecule that is actually a non-protein polymer. It is composed of saccharides and fatty acids. It's a complex sugar polymer known as a xylomelanin.
A sugar antifreeze.
Exactly. It uses a chain of sugars to do the exact same job as the complex beta-solenoid proteins. It proves, unequivocally, that nature found multiple, completely independent, radically different biochemical pathways to fight freezing.
Evolution isn't restricted to just rearranging amino acids into proteins. It can literally build a highly effective, hyperactive ice-blocking shield out of complex sugars and fats.
That is staggering to me. It really makes you wonder what else is out there in the extreme environments of our planet that we simply haven't discovered yet. What other bizarre molecular combinations are keeping bugs alive in Siberia or the deep Arctic?
But whether it's a sugar polymer like the xylomelanin, or a heavily armored beta-solenoid protein in a beetle, or a microscopic zipper in a flounder, it begs the ultimate mechanical question. How does it physically work?
How does a tiny microscopic molecule actually grab onto something as slippery, as rigid, and as dynamically growing as an ice crystal?
It all comes down to a process that scientists call the adsorption inhibition mechanism. To truly visualize this, you have to understand how ice naturally grows at a microscopic level.
When water begins to freeze in a solution, the water molecules slow down and form a lattice. These nascent ice crystals normally exhibit what crystallographers call basal and prism faces. They tend to grow outward as round, flat disks.
Okay, so picture a microscopic, dynamically growing hockey puck.
Exactly.
Yeah.
The hockey puck wants to keep expanding outward, adding water molecules to its edges. What the antifreeze proteins do is they target and adsorb, meaning they physically adhere to the surface of the non-basal planes of the ice crystal.
By attaching themselves to these specific structural faces of the ice lattice, they create physical roadblocks. They block the crystal from growing in its thermodynamically favored direction.
It's like trying to build a brick wall and someone keeps gluing unstackable steel plates randomly onto your bricks. You just can't keep building.
But how do the proteins actually stick? Ice is incredibly slippery. Because initially, according to the sources, scientists thought it was all about hydrogen bonding.
Like the proteins were tiny molecular magnets locking onto the oxygen and hydrogen atoms in the water molecules of the ice.
That was indeed the prevailing, widely accepted theory proposed by Raymond and de Vries, the pioneer we mentioned earlier, back in 1977. It made intuitive sense.
However, biology and physical chemistry are rarely that simple. Later researchers decided to rigorously test this hydrogen bonding theory by purposefully mutating the proteins in the lab.
They systematically altered or removed the specific parts of the protein that were supposed to be facilitating this magnetic-like hydrogen bonding.
If the 1977 theory held up perfectly, the mutated proteins should have completely failed to bind to the ice. They should have been useless.
But they weren't. The hypothesized massive decrease in antifreeze activity was not observed. The mutated proteins still stopped the ice.
Which means hydrogen bonding wasn't the main anchor holding them there. So if it's not a magnetic attraction, what is actually holding them to this slippery ice?
The current scientific consensus, derived from decades of intense study, X-ray crystallography, and advanced molecular modeling programs, points to a highly complex mix of interactions.
Van der Waals forces, which are very weak, distance-dependent intermolecular forces, play a significant role. Along with that is surface complementarity.
Essentially, the physical shape of the protein flawlessly matches the microscopic physical topography of the ice crystal, locking together like an impossibly complex puzzle piece.
But the primary driving force, the main contributor, seems to be largely hydrophobic interactions.
Hydrophobic meaning water-repelling. So the protein basically uses its water-repelling side to forcefully push away the liquid water in the blood, which effectively forces its other flat complementary side perfectly against the solid ice matrix.
Yes. And we can look back at the type I AFPs from the winter flounder as a prime, easily visualizable example of this micromechanic in action. Because of its amphipathic, two-faced structure, it acts quite literally like a microscopic zipper.
The hydroxyl groups of the 4 critical threonine residues on the flat side of the protein align perfectly to bind to the oxygen atoms in the ice lattice along a very specific directional plane.
It literally zips itself onto the ice nucleation structure. Once zipped on, it creates a rigid barrier, subsequently retarding the growth of the pyramidal planes of the ice and depressing the freeze point.
A microscopic zipper, driven by water repulsion, holding a growing ice crystal completely hostage. It's almost poetry. But our sources also tell us that these biological zippers don't always work entirely alone.
Sometimes they bring friends to the fight to make sure the ice stays locked down.
Yes, in the literature we call these secondary molecules enhancers. In beetles, for example, the highly hyperactive antifreeze proteins are often accompanied by other molecules circulating in their hemolymph, their equivalent of blood.
These include polycarboxylates, specifically citrate and other compounds called thaumatin-like proteins. These enhancers interact with the AFPs and dramatically, exponentially increase their thermal hysteresis activity.
They act as an amplifier. They boost the signal of the freeze protection.
Exactly. And the biochemical machinery involved in this whole process is incredibly, unimaginably delicate. Researchers looked closely at the dendroids beetle's highly evolved antifreeze protein, and in the lab, they chemically modified a single solitary amino acid, arginine.
They just tweaked one tiny part of the structure.
That one microscopic modification resulted in the complete and total loss of the protein's thermal hysteresis ability. It was instantly rendered completely useless.
It proves how highly specific, how unimaginably fine-tuned this evolutionary lock-and-key mechanism really is. You change one tiny pin in the lock, and the whole door just flies open to the freezing cold. The beetle dies. It's a game of absolute microscopic precision.
And speaking of fine-tuned mechanisms, I have to mention a genuinely mind-bending study from 2010 that was detailed in our notes, because we keep calling them antifreeze proteins.
But scientists discovered that AFPs don't just inhibit freezing. Because they bind so stably, so perfectly to the ice matrix, they can actually inhibit melting.
In a laboratory setting, they were able to create superheated water ice crystals. They created ice that physically stays solid at temperatures where it should technically, thermodynamically, be liquid water.
Which is a phenomenal physical feat. It perfectly illustrates why the scientific community is shifting its terminology. These proteins are not just simple antifreeze solutes. They are truly profoundly ice-structuring proteins.
They actively dictate and manipulate the structural integrity of ice in both directions, preventing it from forming when it should, and conversely, preventing it from dismantling and melting when it should.
And that concept, the ability to physically structure and control ice, provides the perfect transition to the final major area of our deep dive today. Because once humanity discovers a biological cheat code as powerful as this, we are absolutely, without a doubt, going to figure out how to harness it.
First, let's talk about that name change. There has been a significant PR rebrand in the commercial space. You will notice that food scientists and massive corporations are pushing hard to rename AFPs to ice-structuring proteins, or ISPs.
Why? Because when average, everyday consumers read the word antifreeze on the back of their food packaging, their mind immediately, understandably, goes to that toxic neon ethylene glycol in their car radiator.
Even though the biological proteins and the car chemicals have absolutely nothing in common besides their end goal of managing ice, the name antifreeze is a complete nightmare for marketing.
It is a very necessary distinction for public understanding and commercial acceptance. Because, as you alluded to, the commercial applications of these proteins are not some far-off sci-fi future. They're already here, right now.
So what does this all mean for you? You might actually be eating these highly evolved ice-structuring proteins right now.
Indeed. The multinational consumer goods company Unilever recognized the immense multi-billion-dollar potential of these ice-structuring proteins for the global food industry, specifically the frozen dessert sector.
Years ago, they underwent rigorous testing and secured official approval across the United States, the UK, the European Union, China, Australia, and many other nations to use these exact proteins in commercial ice cream production.
They utilize a highly specific, genetically modified yeast to safely produce the exact same antifreeze proteins originally found in those freezing deep-sea fish. And they put them directly into mass-market products, like popular popsicles and the highly successful Breyers Light double-churned ice cream bars.
Okay, but taking a step back, why? Why on earth does my vanilla ice cream need fish antifreeze in it?
To solve the absolute greatest enemy of the commercial ice cream industry, temperature fluctuation. Let's trace the journey of ice cream. When it is manufactured, it is perfectly smooth.
But then it has to be transported. It might sit on a slightly warm loading dock for 20 minutes. It goes into a delivery truck. It goes into the grocery store freezer, which goes through defrost cycles.
Then it goes into your grocery cart as you walk around the store. Then it thaws slightly on your kitchen counter before you finally put it back into your home freezer.
During every single one of those slight temperature fluctuations, the microscopic, smooth ice crystals inside the ice cream melt slightly and then refreeze.
And when they refreeze, they undergo that exact same recrystallization process we talked about earlier with the freeze-tolerant plants and bugs. Thermodynamics forces those tiny crystals to fuse into larger, jagged, crunchy ice crystals.
Which completely ruins the texture. It gets that weird, icy, crunchy, freezer-burnt texture that everyone hates.
Exactly. But by adding these ice-structuring proteins into the mix, Unilever completely controls the ice crystal growth during those inevitable temperature fluctuations.
Just like in the freeze-tolerant organisms, the AFPs bind to the tiny ice crystals the moment they start to refreeze, and they physically stop them from growing larger.
This structural control allows Unilever to produce incredibly dense, rich, creamy, and notably reduced fat ice cream with far fewer artificial chemical additives, because the biological protein is doing all the heavy lifting of maintaining the smooth microscopic texture.
It is brilliant engineering. Now, whenever we mention genetically modified yeast and the insertion of fish genes into mass-market food, it is important that we impartially address the concerns noted in our reading.
There are anti-GMO organizations and consumer advocacy groups that have raised concerns, suggesting that introducing these novel antifreeze proteins might cause inflammation or allergic reactions in humans.
It is a concern that has been raised publicly, yes. However, analyzing the data strictly from an objective toxicological and historical standpoint, the human intake of these specific AFPs in our diet is already quite substantial and has been for millennia.
Historically, humans living in temperate and northern regions have consumed these exact proteins naturally for centuries. Every single time they eat a cold water fish, winter-harvested vegetables, or cold-tolerant edible mushrooms.
Based on this extensive history of natural human consumption, the overwhelming scientific and regulatory consensus is that their functional ice-binding properties do not impart any toxicological or allergenic effects on the human body.
And as for the transgenic yeast process itself, using a modified yeast to brew the protein, the source material is very clear that this is standard, deeply established practice in the modern food and medical industries.
This is the exact identical biotechnology used to produce modern insulin for diabetics. It is the exact same process used to produce the rennet that is used to make almost all modern mass-produced cheese.
You isolate the specific gene you want, you insert it into a fast-growing yeast, and you let the yeast act as a microscopic brewery to safely, cleanly produce the protein in massive vats.
And from an ecological and sustainability standpoint, producing it in yeast is vastly, immeasurably superior. It makes industrial production highly efficient, and more importantly, it absolutely prevents the industry from having to harvest and kill thousands, if not millions, of deep-sea fish just to extract trace amounts of the protein from their blood.
It's a win-win. We save the ocean ecosystems, and we get perfectly smooth ice cream that survives the trip home from the grocery store.
But perfecting the texture of dessert is really just the tip of the iceberg, pun fully, unabashedly intended, because the future commercial and medical applications currently being researched are massive.
We are looking at agricultural revolutions, increasing the freeze tolerance of vital crop plants to extend harvest seasons and protect against devastating sudden frosts, improving farm fish production in much colder climates to ensure global food security,
Lengthening the shelf life and nutritional integrity of frozen foods worldwide, improving the pinpoint precision of cryosurgery, where doctors use extreme cold to target and destroy cancer cells.
The agricultural and surgical applications are incredible, but then there are the absolute holy grails of cryomedicine. We are talking about the ability to dramatically enhance the preservation of human organs and delicate tissues for transplants.
Right now, as you may know, the window to transport a donated heart or liver is incredibly frustratingly short. You cannot freeze a donor organ because the resulting ice crystals will immediately rupture the cell membranes and destroy the tissue beyond repair.
If we can master the use of synthesized AFPs to safely freeze, transport, and then flawlessly thaw human organs without cellular damage, it would fundamentally revolutionize global transplant medicine.
Beyond that, there is active research into advanced therapies for severe hypothermia, and potentially the ultimate, most ambitious frontier of all, human cryopreservation, often referred to as cryonics.
The theoretical ability to safely freeze an entire human body, halt all biological degradation, and revive it decades or centuries later without massive, irreversible cellular damage.
It sounds like pure science fiction, the stuff of space operas and futuristic movies, but the biological blueprint, the exact molecular mechanism to make it happen, already exists. It has been perfected. It is floating right now in the blood of an Antarctic fish and the hemolymph of an Alaskan beetle.
To quickly recap the incredible mind-expanding journey we've been on today, we started with deep-sea drilling cores revealing a 30-million-year-old climate catastrophe, a mass extinction that forcefully pushed surviving fish to blindly evolve their own digestive enzymes into a biological antifreeze.
We learned about smelt pulling off an impossible evolutionary heist to steal survival DNA directly from herring. We scaled all the way down to microscopic diatoms and edible fungi surviving the brutal sea ice, and we looked at heavily armored bugs that can comfortably, casually survive negative 30 degrees Celsius.
We decoded the complex microscopic zipper mechanisms and hydrophobic forces that physically lock down expanding ice crystals. And finally, we traced how all of this ancient evolutionary brilliance has been captured by modern biotechnology to keep your Breyer's ice cream perfectly smooth.
It is a profound, humbling demonstration of nature's relentless adaptability. But there is one final, entirely unmentioned thread to pull from our source material that I want to leave you with today. Up until very recently, we were entirely dependent on biological sources. But in 2021, a breakthrough occurred. Scientists at the EPFL in Switzerland and the University of Warwick successfully created an entirely artificial imitation of antifreeze proteins in the lab.
We are no longer strictly reliant on harvesting or brewing nature's designs. Human science can now actively synthesize these ice-structuring mechanisms from scratch.
Furthermore, consider the Alaskan beetle we discussed, the one that completely bypassed proteins and uses a non-protein sugar polymer, xylemanin, to easily survive the freezing tundra. This convergence of synthetic biology and non-protein cryoprotectants raises a highly provocative question.
If humans can now artificially synthesize these complex ice-structuring mechanisms, and if nature has already proven that you don't even need complex, fragile proteins to do it, what does this mean for the future of human space exploration?
Could we synthetically adapt human biology, or perhaps the delicate crops we need to sustain us, to comfortably survive the deeply frozen, hostile environments of Mars, or the icy, radiation-blasted moons of Jupiter like Europa? Could an artificial, mass-synthesized xylemanin be the key biological shield that eventually protects the first human colonists from the lethal subzero vacuum of deep space?
That is an incredible, profound thought to end on. From a small fish swimming in the dark, freezing waters of an ancient Antarctica, to the very real potential for human survival on the frozen surface of another planet. Thank you so much for joining us on this deep dive. I guarantee you're going to look at the winter frost on your window and your next bowl of vanilla ice cream in a completely different light.